Okay, so somewhere around 3.5 to 3.8 billion years ago, we see our first prokaryotic cells, right? And they're coming from those protobionts. So what is a prokaryotic cell first? So a prokaryotic cell, and kind of the name, pro and karyotic. So this term here, karyo, means nuclear or nucleus. Pro means often means before, so it's a cell without a nucleus. So we have the cell membrane, and we'll get into prokaryotic cell structure in specific lectures on prokaryotic cells, on bacteria and archaea. This is sort of more a little introduction to their origin and other things that happened along the timeline, because that's kind of what we're doing now, is working along the timeline. We'll come back to some of these things later to get into them in more detail. So this isn't everything about prokaryotes we're going to cover. In addition to a membrane, prokaryotic cells are going to also have a cell wall, which again is something we'll focus on a little bit more later on. But the membrane's the same as our membrane's a phospholipid bilayer, made up of the same types of molecules. Archaea, another group of prokaryotes, their phospholipids are a little bit different in terms of their structure, but not like ours. They're something a little more unique in general. And so is their cell wall, but same basic sorts of structures. Inside they have DNA, so that's their genome. Now the DNA, so the DNA genome of the organism is double-stranded DNA. So there's two strands of DNA, just like our DNA. One of the differences, though, is that this DNA, and this is going to come back to be important later on, is circular. So circular DNA means, our DNA is linear. So linear DNA means it has ends too. So there are two strands, there are bonds between the strands. One strand ends in a phosphate, this ends here in an OH group, then this strand here ends in a, well, that one also ends in a phosphate because the way I did that. But each strand, this one has a OH group here. If we were to follow it, at the other end you can see there's a phosphate, like that. With the bacterial cells, or the prokaryotic cells, their DNA is circular, so there are no ends. The ends are then joined or linked together into a loop, right? And that's important or significant as a characteristic. We don't have that in our cells, and the prokaryotic cells do. So there's no nucleus, so there's no membrane around this. It's just free in the cytoplasm. So this is all water and other molecules, a cytoplasm called the cytosol. And that's where all these reactions take place, and that's the basics. And then there's a lot more details and all to it, but that's the idea. So these organisms came about around this time. So how do we know? So evidence, because they're really small. So we think about evidence, fossil evidence of a dinosaur, which is gigantic and is sometimes difficult to find. How are you going to find evidence of bacteria, which is much smaller than our cells, so difficult to see under the microscope? You need a much more higher magnification to even see them at all, and they'll still look very small. So the fossil evidence we have and continue to find, are for structures that are formed by bacterial films. So bacteria, once they start to live, they don't just live as a single cell just floating around by themselves. A lot of them join with other bacteria and they start to form mats, right? So there's like layers and layers, like coatings of bacteria. And these are called bacterial films. And some of these bacterial films, which then live on, in, and around sediments, became fossilized. And these fossilized remains are called stromatolites. And the stromatolite remains, we find them, around this time period. Okay, and so we know that there's evidence that some bacteria had to exist even before this time period, so that they could form, more complex associations with one another to form these films and leave these fossil remains behind, right? From groups of them. So again, not the single cells, but the groups of them. And so now we know that they came about. ultimately, what we're trying to do is get here. Like I said, we're not going to focus on the prokaryotes. We're focusing more on the timeline right now. So the idea for what we're going to cover here is to go from the prokaryotes to the eukaryotes, and what happened in between. Okay, so we're looking at over another billion years, and so somewhere in this time, something else starts to happen. Previously, we talked about molecules and the synthesis of new molecules and so on, about the chemicals in the Earth's atmosphere and how they would interact with one another and maybe form new molecules. Well, one type of molecule that was actually much lower in the atmosphere than it is right now is oxygen. There wasn't much oxygen at all in the Earth's atmosphere. Most all of our oxygen is biologically produced. It comes from the process of photosynthesis. So before we really get into this, without going into all the details of photosynthesis, you should know something about photosynthesis and what it is. So in the process of photosynthesis, what we have is light, so energy from light, being absorbed by pigment molecules. And these are the pigments. So you can imagine a plant, a leaf is green, and because there are these green pigments and a whole variety of other different pigments. And when the light is absorbed by these pigment molecules, electrons get excited. So this is an excited electron. Again, we're not going into all the details of the process, but there's a little bit you should know for some of this to actually make sense, for the main point to really make sense. So excited electrons can jump up and they can fall back down and they can pass on energy, or the excited electrons can be grabbed by proteins and then passed on to an electron transport chain, which hopefully most of you at least have heard of if you don't know all the details of already. So that means these molecules, these pigment molecules, become oxidized, which means they lose electrons. And those electrons go into an electron transport chain. Some of it could be used to pump protons, which could then be used to store energy, which could then be used to make ATP, and can then be used to make other molecules called NADPH. So ultimately, the things that are made here are ATP and NADPH, and that's a whole separate thing when we go into what is photosynthesis, how does photosynthesis work, and all the details. The main thing we want to focus on here is what happened here. Some molecule here, a reaction center chlorophyll, has lost electrons, so it means it's been altered chemically, and now it's not going to be able to function anymore. So there are some of these that have the electrons replenished by other pigments. As they are passed through electron transport chains, they actually go to others, and then they can kind of move on. So they always have a supply. But some of them don't have a direct link to it. They need to get the electrons from somewhere else. So in comes water. So water, we have oxygen and hydrogen. What's going to happen here is, each hydrogen is one electron and one proton. And so the water molecules will come in here, and there's a special enzyme that will then break off the hydrogens from the oxygen, and it'll then split the hydrogens into their proton and electron form, and it'll use those electrons to replace the lost electrons. So essentially, this is part of photosynthesis, is to split water so that you can get electrons to keep replenishing the electron transport chain and the photosynthesis process. So photosynthesis occurs, and it's going to continue to occur. It's going to need this electron supply, and it's going to come from water. So the hydrogens are kind of being used up. What's left are oxygens. These oxygens will then join with other individual oxygen atoms to form molecular oxygen. That molecular oxygen, the oxygen that you're breathing, came from this process, right? And so did the oxygen way back, billions of years ago in the Earth's atmosphere. And the levels continue to rise and rise and rise till around the levels that they even are today, during this time period. And that's coming from bacteria, right, or bacteria that we have today that do photosynthesis. There are a type of bacteria called cyanobacteria. And so it's likely a cyanobacteria ancestor. So we have cyanobacteria. This is a current modern day bacteria that is photosynthetic, that does photosynthesis. Ancestral organisms of these cells were in the Earth's oceans doing photosynthesis. Again, what kind of evidence do we have for this? Again, some of the gas pocket evidence, but also we have changes in oxidation of iron. So some of the Earth's, so we kind of have these rust lines in the ocean. So this rusting occurred around 2.7, this is this time right here, around 2.7 billion years ago. We know this oxygen is in the atmosphere because of these rust bands. In iron-containing rocks that could be dated back to about that time period. Right? And so the process occurred in the oceans. The cyanobacteria or their relatives produced it there. Eventually, as the oceans and the water became saturated with oxygen, it then left. So that means before this time period, so it's not before the 2.7 billion years ago, this was going on. So this was going on from much earlier, right, in the past. It's just around this point is when it really started then coming up into the atmosphere, and that started changing a lot of things. So it changed the organisms themselves, because oxygen itself can be an oxidizing agent, which means it can help pull electrons away from things, which means they can be damaged. So at this point, this new oxygen, which we're thinking of as good because it's something that we need, is actually bad, right, for any living thing. Because most living things at this particular time, which are other prokaryotic cells, are what we call anaerobic, which means they operate in the absence of oxygen. So aerobic means with oxygen or using oxygen. Anaerobic means they don't use oxygen. So we have some bacteria today that are strict anaerobes, meaning that if they're exposed to oxygen, it actually kills them. Others are facultative anaerobes. So they could be in the presence of oxygen, it doesn't kill them, but they're not using the oxygen necessarily. Or they can sometimes alternate between different types of metabolism. But at this point, since there was no oxygen, there was no reason to use the oxygen, so most of those organisms didn't exist, and they were most likely stressed by this oxygen, potentially killed off, and this could have caused one of the first mass extinctions of prokaryotes, the ones that were anaerobic. However, with variation, right, in populations, there would be some that could either tolerate the oxygen, and it didn't bother them, because they had other variations to some of their molecules to protect them, or they could even use the oxygen. So now, after we get photosynthesis occurring, what could start to happen is this. We have some bacterial cell which is anaerobic, can't use oxygen, and then we have another bacterial cell who's aerobic and can use oxygen. This cell here, maybe just as part of its normal habit, just consumes or phagocytizes other cells. So, and it could consume any cell. It's not consuming the cell necessarily because it's aerobic. It's just consuming it because it's another cell, and this cell eats other cells. But what it finds out is that after consuming or eating this cell, something happens. It essentially feels better. It's less stressed. This cell that's inside it is binding to this oxygen, and it's pulling it away, and so it's not harming this particular cell. Potentially, this cell is also giving off, so it might be taking away stress or waste from this cell, and it might be giving off some kind of nutrients that it can produce that this other cell gains. So potentially it just destroys it, but over time other organisms consume other organisms like this, and some of them then don't destroy this other cell that they ate, they keep it right inside them. And over time they start to develop a relationship, a symbiosis. What they both benefit. This cell here, the one that was consumed, actually might gain protection or some other benefit, maybe just a constant nutrient supply from the host cell. The host cell is gaining a benefit because this other cell is maybe detoxifying the oxygen and or producing other molecules that it can use as well for its metabolism, so they kind of have a back and forth. So this idea is called serial, sequential endosymbiosis. And in many ways it's not just an idea, so because we refer to this as serial endosymbiosis theory, and what you need to know, you should know by now, but if not, you need to. In science, the word theory is an accepted explanation for a natural phenomena that is supported by evidence. It's not just an idea. So it's something that we use to explain some very complex phenomena. We continually gather data and information on this, but we have information that actually supports it. A hypothesis is where we just have an idea and we may not yet have all the information to support it or accept it as an explanation. But a theory is the accepted explanation. So serial endosymbiosis is what we accept is how cells that were prokaryotic came together to form our eukaryotic cells. And one of the reasons why, the driving force behind it, is the increase in oxygen in the atmosphere. So let's talk about some evidence, or examples of how this potentially happened, or why it happened, or can we show that this actually could happen? Well, there are a number of things that we can look at that can point to this being the way that it actually occurs. So if we look at one of our cells, a human cell, and we have our own nucleus with our DNA in it and so on, and we have within ourselves mitochondria. I'm not going to draw a full mitochondria here, but if we look at a mitochondria, blow it up a little bit, mitochondria are a double membrane structure. So if we look at this, if we think of this process, okay, if this cell ate the other cell, this is the cell's membrane here, and then the cell that it consumed had its own membrane, so now they're 1-2 membranes, so it's a double membrane structure. So we have that. Second thing we have is if we look at our mitochondria, inside the mitochondria, we find something interesting. Mitochondria have their own DNA. That's important. So proteins that are within the mitochondria, ribosomes inside the mitochondria of your cells, and your cells don't just have a single mitochondria, your cells have hundreds, potentially thousands, and it varies depending on the cell type and how metabolically active it is. So let's say muscle tissue is going to have a lot more mitochondria in them than a fat cell is going to have. So they're not all exactly the same, but there's more than one. There's a lot of mitochondria in our cells, and every mitochondria has its own DNA that is separate from the DNA in your chromosome. It's not the same. This is eukaryotic DNA, and this DNA is prokaryotic. Why would we say that? Well, because it's a circular DNA, and it has the ribosomes produced from it are more similar to the prokaryotic ribosomes than eukaryotic ribosomes produced by our chromosomal DNA. So we have its own DNA. They code for their own genes. Their structure of their genetic material is like prokaryotes, and many of the genes that they express are more similar to prokaryotic structures than they are to our today modern eukaryotic structures. So this gives supporting evidence to this theory that our organelles, like mitochondria, for example, came from symbiotic relationships between prokaryotic organisms. What's the job of the mitochondria? Well, this is where oxygen goes, right? Oxygen goes, that's where all your oxygen goes. It's inside your mitochondria. There's an electron acceptor for the electron transport chain, and it binds to oxygen, and then the oxygen takes those electrons from the electron transport chain. That's kind of the job of oxygen, is to receive them, and that's all happening here at the mitochondria. So we have this bit of information. We also have examples of some organisms that are symbiotes or parasites, cellular parasites. So some cells that actually may go the other way, where instead of being eaten, there's one cell actually, this cell might parasitize the other cell. So this cell comes inside this cell and lives inside it. So if it parasitizes it, it hurts the cell, But we have some examples that are known of an archaea, for example, called a group called the Nanoarchaeota. The Nanoarchaeota are a group of archaea, so they're prokaryotic cells that are so simplistic in their genome that they can't express a number of genes that they need to survive on their own. So they can't even survive on their own. They actually have to live inside another cell. They live inside another prokaryotic cell, another bacterial cell. So we have a bacteria that lives essentially inside a bacteria. And we have this as an example today. Some parasites, like Rickettsia, that's what they do. They parasitize eukaryotic cells, but we see some cells then living inside other cells. And so this, again, idea is not completely foreign. We have organisms like corals that are animals that undergo photosynthesis. They don't have a photosystem to do photosynthesis, but they have symbiotic cells, algal cells, living inside their cells that run photosynthesis. So this is why we see or hear things called coral bleaching. That essentially means all those little algal cells that live symbiotically inside the corals are expelled, right, and released, and so the corals lose the pigment, and they lose the color, and they lose the ability to get energy from those algae that are running photosynthesis, called zooxanthellae, and they become stressed, and they may die, or they may not die. They might recover, but it's not particularly good for them. So it's a symbiotic relationship. It's a good thing for the corals to have these cells living inside their cells. So symbiosis is something that we see a lot of in nature, and so the idea of the endosymbiotic explanation is highly likely, right? And it's supported by some molecular evidence, different types of genes, and some of these other examples. And so then we get eukaryotic cells around 2 billion years ago, 2.1 billion years ago, coming about most likely through this process. And now we have something similar happening with chloroplasts, where chloroplast was most likely a cyanobacteria that was then consumed by another cell. Now this cell, like the zooxanthellae, have the ability to gain more energy from the photosynthesis process of the other cell. So they don't have the ability to run photosynthesis, but they can reap the benefits of it because they have a symbiont who does. And chloroplasts are a multiple membrane structure. Chloroplasts have their own DNA. It's circular. It's prokaryotic DNA. So mitochondria and chloroplasts are organelles in eukaryotic cells that likely came about separately, but along similar pathways, similar mechanisms were used at their origins. And so that's kind of the next step, in this is kind of like, how did the eukaryotic cells come about? And our explanation for this is called serial endosymbiosis theory, often referred to as SET. So if you just see this S.E.T, it's referring to this idea. So have the basic dates. Again, these dates are rough, all right? But you should have the idea on the timeline where or when these things are happening, and some of the terminology introduced here. So the aerobic, using oxygen, anaerobic, without oxygen, the process of the serial endosymbiosis, and a little bit about the driving force from photosynthesis, which requires oxygen to be released because electrons are being taken from water. So you should look at that a little bit. You don't have to know all the chemical details of it, but you should be familiar with it because that is the driving force behind the process. We will go into more details on prokaryotes, but at this point you could get the circular DNA as a characteristic and no nucleus, as a characteristic, and as we'll add to those characteristics in the future.